Genetic diseases have plagued humanity for centuries. Today, we stand at the cusp of a new era, one where we can rewrite the very code of life, thanks to CRISPR. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing tool. Discovered in bacteria as a defense mechanism against viruses, CRISPR has quickly become a cornerstone of genetic research. Its simplicity, precision, and versatility have opened up unprecedented possibilities for treating diseases and engineering life itself. CRISPR has democratized gene editing, making it faster, cheaper, and accessible to researchers worldwide. CRISPR is an elegant system composed of a few key components working together to edit DNA. At the heart of CRISPR is the Cas9 enzyme, the molecular scissors responsible for cutting DNA. This enzyme is directed to its target by a guide RNA, a short RNA sequence that acts like a GPS navigation system. The guide RNA is designed to match a specific DNA sequence, ensuring that Cas9 cuts at the precise location intended. Once the DNA is cut, the cell's natural repair mechanisms kick in. The repair template is a piece of DNA that carries the desired sequence change. By designing different guide RNAs, scientists can target Cas9 to virtually any gene in the genome. CRISPR's gene editing capabilities extend beyond simply cutting DNA. Gene correction involves correcting a faulty gene, promising for treating inherited genetic disorders. By supplying a repair template with the correct sequence, CRISPR can correct the mutation and restore gene function. Gene disruption involves disabling a problematic gene, explored for diseases like cancer. By disrupting these genes, CRISPR can potentially halt or slow down tumor progression. Gene addition involves inserting a new gene into the genome, holding potential for diseases like type 1 diabetes. CRISPR could be used to insert a functional insulin gene into cells, restoring insulin production. The versatility of CRISPR opens up a world of possibilities, paving the way for personalized medicine. CRISPR's potential extends far beyond medicine, touching upon agriculture, energy production, and our understanding of life. Clinical trials are underway for genetic disorders like sickle cell anemia and cystic fibrosis. CRISPR offers hope for a cure by correcting faulty genes at their root. Beyond genetic disorders, CRISPR is being explored as a potential weapon against cancer. By engineering immune cells to recognize and destroy cancer cells, CRISPR could revolutionize cancer immunotherapy. CRISPR's ability to target and disrupt viral DNA makes it a promising tool for new antiviral therapies. In agriculture, CRISPR can develop crops resistant to pests and diseases, addressing global food security. For energy, CRISPR can engineer microorganisms to produce biofuels more efficiently, paving the way for sustainable solutions. While CRISPR holds immense promise, it's essential to acknowledge the challenges and ethical considerations. One significant challenge is the potential for off-target effects, where Cas9 may cut DNA at unintended sites. Scientists are working on improving CRISPR's accuracy and minimizing off-target effects. Delivery of CRISPR components into desired cells is crucial for therapeutic success. Various methods are being explored, including viral vectors and nanoparticles. The ethical implications of altering the human germline raise profound questions. International guidelines are being developed to ensure the responsible use of CRISPR technology. The future of CRISPR is brimming with possibilities. Research is focused on improving CRISPR's precision and developing new variants of the Cas9 enzyme. Novel CRISPR systems beyond Cas9 are being explored, offering alternative approaches to gene editing. Innovative delivery methods like nanoparticles and engineered viruses are being investigated. CRISPR's applications continue to expand, from new diagnostic tools to climate-resilient crops. CRISPR holds the potential to reshape our world, offering solutions to humanity's most pressing challenges. The journey ahead requires collaboration, innovation, and a commitment to ethical considerations as we unlock CRISPR's full potential.